Okay, and now we're going to move into the chapter number 4, and we're going to talk about gene linkage and genetic mapping. And we're not going to talk only in a theoretical terms. In the lab, we're going to cover all these topics too. Okay, so the class will be divided in 3 main sections. In the beginning, I will talk about genetic linkage, and we will solve exercises like this one. Then we will talk about genetic mapping, and we will cover topics that we start discussing in genetic revolution about Thomas Morgan. Then, in addition to talk in terms of theoretical terms, I will also show you in the lab how to do these 3-point crosses and why is the importance of those things. Okay, so important that you remember that you know that you need to understand or to learn these topics at the end of this chapter. What is a linkage? Remember what is recombination, and now we're talking about that in a more molecular from a more molecular perspective. What are topics like what is centennia, what is coupling and repulsion, and what is the difference? There will be a question of the test to talk about that. Then we will talk about what is this centimorgans and how we used to map chromosomes before and how we do it now, and also what are linkage groups. And in addition to this, we will cover a little bit of what is the nomenclature that we use to describe the genotypes on not only in fruit flies but in other organisms. So we have studied this in so many courses, and I'm sure that you know that when genes are in different chromosomes, we can understand why independent assortment occur. Now the question that we want to discuss is what happens when you have two genes that are in the same chromosome. Yeah, so we're going to talk about crossover, and I wonder you think what happened here when you have this mixing of pieces of chromosomes, and I wonder you think, okay, it this always happen and it happened at the same rate. It happened. What happened with the distance in the chromosome is the same will happen if you have the genes when they are very far, or what happened when they are very close. So we are going to answer those questions today, and also we are going to prove those concepts in the lab using fruit flies. So now we're going to talk about a recombination. Yeah, so it's divided in this word is divided in re and combination. So in other words, is the shuffling of genes. And remember, Wikipedia is wonderful to do some things, but sometimes it's not precise. So I want that you remember that this recombination, the mixing, the creation of genetic variation, is fundamental for both genetics and evolution, and we will see that in prokaryotes they use different mechanisms. They share plasmids, and it's like imagine what we do when we share files in using internet and sending emails to everybody. This is how bacteria can get antibiotic resistance very fast. However, multicellular organisms they cannot share information in that way. We cannot share plasmids, so we have to create generic variation in different ways. And one way is using crossing over. And so there are two ways of define recombination. So one, it could be through independent assortments. You have a male and a female and a sperm and an egg, and then now you are going to mix the information of these two. And so that is a very efficient way. So in other words, uh, while many prokaryotes are asexual, um, many eukaryotes and especially multicellular organisms, they have sexual reproduction, and these allow to create more variation through just mixing of alleles. However, there is another very important component that is called crossing overs, and that we will study in this class, and is that you just take one chromosome and another chromosome, and there are specific enzymes here that allow to cut the pieces of DNA in a specific way and interchange information and create these recombinants. Yeah, that is a gamete with a combination of alleles that didn't come from its parent is a recombinant gamete. So if you see now you have a mix of pieces of chromosomes that it didn't come from its parents, and we will talk more about that in a second. So just from this, I want that you remember that recombination is very important for genetics and evolution. I want that you think that there are two types of ways of shuffling alleles and genes. That one is independent assortment, the second one is crossing over. And in the crossing over, you take a piece of a chromosome and another piece of a chromosome, and you have enzymes that are specialized, that are specific regions that allow that this happen without losing information. And this is a very important mechanism. And we will talk about this word recombinant many times during this class. So remember, every time that we teach you something in an introductory biology course, later we say, okay, but this is not as simple. So independent assortment, great tool to study to create variation. However, as I told you before, there is a process that happens during meiosis in gametes that the homologous chromosomes trade pieces with each other. And so now the gametes, if you see, all these gametes have pieces from another chromosome, and these are called recombinants. So all these all mixed forms are called recombinants. So every time that we did a Mendelian cross and we have information from a female gamete and from a male gamete, and now they will be together, actually what happened is not only that you have that, is that in some cases there will be parts of the DNA that they can get broken and they get stuck. So as a result, you're going to have regions, gametes that they will have no recombination. Yeah, so they will look exactly the same as the gametes from the parent. So they will be non-crossover and non-recombinant. And at the same time you will have other ones that you will have combinations, yeah, that you will have. So this one is only red, yeah. However, in the other two, these are represent recombinants because there was crossover in that. So in addition to what we learned with Mendel, something more complicated can happen in some cases, and the gametes will not look exactly the same, the chromosomes. They could be a combination of pieces of chromosome, and we call that recombinant. But how do we tell? How do we know that the crossing over is actually happening? And I know the answer that we are thinking, all of us, is, Nicholas, this is very easy. You just need to sequence the DNA and you can see clearly if there was a piece of a chromosome that attached to another one. And right now, this is relatively easy to do, and this is how we test if there was a recombination or no. However, this phenomena was discovered before gene sequencing was developed. What they did in the past is they took alleles located near to genetic markers along the chromosome. So let me show you here, for example, using an example of fruit flies. So here, imagine that this is an allele A, that is, they have tiny antenna, and here this is B, that the flies have an ebony body, a black body. Well, here you will have this long arista and this gray body that it will be B. So if you find that there is no recombination, you will find some of these ones will look like this, while other ones will look like this. So this is perfect. There is no problem. But what about when you find that some of them start looking slightly different, and you find some phenotypes that have that they look like this. Yeah, that you have flies that have the short antenna, but also they have gray body. So that was not like the parentals. So they was a recombination event. And what about if you also find the opposite, that you find flies that have the long antenna, but at the same time the ebony body, the black body, like what happened here. So in this case, you can think and infer that there was a piece of the chromosome that mixed with the other one, and this is how they used to do it. And this is how we will do it using using this type of tools, using these markers, using fruit flies in the lab. So what they found is that, I want that you remember this, that there will be sections of the chromosome that there will be definitely no recombination. There will be sections of the chromosomes that there will be definitely recombination, and there will be sections of the chromosome that some genes that they will be independent assortment. Yeah, and what is the key difference between these three regions? This between these three behaviors, in many cases, is just the distance. Yeah, so if the distance is too short, there will be no recombination. So if this is short, is too is there is no recombination. If the distance is long, if there are in two different arms of the chromosome, they will have they will behave like if they were in different chromosomes. So they will independent assortment will work, and if they have an ideal distance, they will be different degrees of recombination. And we will talk about that in the following slides.